Hello all, my name is Hampton Hand and I am a genetics undergraduate student at NC State. The field of genetics I find to be simply satisfying, as we are the people we are today due to the combination of amino acids, A's, T's, C's, and G's that make up our DNA. The field of genetics has been very prevalent in the means of Alzheimer's disease, now that we have realized that it is one of the leading factors of the disease. For many years, the treatment of Alzheimer's has been heavily debated and looked at. However, there has been no success. However, now, through the use of nanotechnology, there could be a treatment for this developmental disease. Therefore, I would like to present to you the use of nanotechnology for Alzheimer's research as an evolutionary step in modern medicine. Treatment of neurodegenerative disorder, Alzheimer's, has been scrutinized for several decades with little to no progress or success. It is pondered as to why there seems to be no permanent response to a disorder affecting about 5.8 million individuals of all ages within the United States. So why has this research not been prevalent in the medical and scientific community? While modern-day medicine has allowed professionals to build a better understanding of the body, the mind still holds a lot of unknown factors, chemicals, and proteins that make up complex systems that we are yet to be enlightened on. However, it is time for these statistics to disappear. A particular field of research that has remained uninvestigated until now would be that of genetics. The department analyzes the smallest variations in DNA and how they can lead to life-changing effects on the human body, in this case, Alzheimer's. The permanent change or mutation in one or more specific genes has been thought to cause diseases. Diagnosis of both early onset and late onset Alzheimer's is said to be due to a genetic factor, leading to the onset and neurodegeneration of individuals. As shown, this only increases the growing epidemic of Alzheimer's disease, but it makes sense that we should search for a treatment more desirable. Nanomedicine has shown promising potential as a modern-day medical treatment to combat Alzheimer's epidemic. Nanomedicine refers to substances where nanotechnology is combined with drugs and diagnostic molecules, which allow for the specific targeting of cells and tissues. Previous research has shown that lipid-based nanoparticles and carriers and the use of delivery of Alzheimer's therapy through nasal routes have resulted in permanent neurodegeneration management, suggesting that the use of different delivery methods of nanomedicines would result in complete restoration of the brain if successful in crossing the blood-brain barrier. This is where many researchers have complications for both invasive, which is the brain, and non-invasive delivery, which is the nose. So, why don't we investigate that now? With a better understanding of the brain, and now that nanomedicine stands at its own caliber, the impact of finding a cure that could later be used in other central nervous system diseases treatment would not only save thousands of lives, but also change modern medicine significantly. This simply would be revolutionary. So why am I passionate about this subject so much? Why do I want to research this? Personally, I have seen the effects on Alzheimer's on loved ones and have been involved in previous research. However, I am ready for a cure to be out, and I think this would be the leading steps. So what are my research objectives? I want to build on the challenges previous researchers have expressed through examining the form of delivery of nanomedicines and anti-Alzheimer's disease drugs by analyzing which method is more successful in passing the blood-brain barrier. I want to manage and terminate previous neurodegeneration and determine which nanostructures or nanoparticles work the most effectively. Many individuals today are affected by Alzheimer's disease. The use of nanotechnology can effectively carry and deliver drugs to the brain. The research debates the different routes to administer the nano devices to target the brain, bypassing the blood-brain barrier. To determine which method will best resolve the neurodegeneration, we will investigate the difference in both non-invasive and invasive forms of administering anti-Alzheimer's drugs through the following research. This would enhance bioavailability and maximize pharmacy therapy within Alzheimer's patients. This coming year, many patients have been registered for nanotechnology-based projects. However, clinical trials have not yet occurred, and it is now time for one to happen. But keep coming back to this idea of genetics being one of the causes of Alzheimer's that we now have realized. Mutations in presenilins and amyloid B are associated with early onset Alzheimer's. Conversely, mutations in the gene encoding apolipoprotein E4 or POLE-E4 are associated with late onset. This is what causes the neurodegeneration. Another idea that I keep coming to is the blood-brain barrier. The blood-brain barrier, or BBB, is the structure responsible for protecting the brain, acting as a gateway against the circulating toxins and cells through a selective permeability system. Hence, the delivery system for the treatment of brain diseases should have the capacity to cross the blood-brain barrier level without causing immune responses. However, this is a challenge for the delivery of pharmaceutical drugs to the brain. And the trick? If there is dysfunction of the blood-brain barrier, it can trigger oxidative stress that promotes amyloid B generation. This causes a feedback loop that causes cognitive impairment. Through the regulation of nanotechnology, however, Alzheimer's can be treated. Development of nanotechnology, however, has resulted in incredible insights into the function of the complex neural activity of the brain and approaches that diagnose and therapy of brain diseases. The reduced size, biocompatibility, prolonged blood circulation, and non-toxicity of nanomaterials have created an upcoming delivery system that can transport the therapeutic agents to the brain in a simple manner. The perks of this? The nanotechnology-mediated drug delivery systems are based on both specific and non-specific mechanisms for targeting brain sites. My research will be looking at both a non-invasive and an invasive form of delivery of therapeutic agents to the brain. The invasive route involves the surgical administration of drugs directly inside the brain, which must provide satisfactory dosage without causing harm to other regions of the body. However, the amount of the injection relies on the cerebral diffusion, which is a concentration dependent and can cause a decrease from the administration site. On the other hand, the nasal route is preferred over the systematic drug delivery, as the drug is directly delivered into the brain through the olfactory bulb, which increases the bioavailability and reduces degradation of the drug. However, limitations such as poor drug permeation through the nose and clearance might be encountered. Therefore, each form of the drug delivery must further be studied to analyze which would be the most effective. So my big question is, what form of drug delivery is most effective when it comes to anti-Alzheimer drug delivery? This experiment must ultimately be a clinical trial using the species humans. While this would need to be ethically checked, Alzheimer's drugs do not have the same effect on lab species such as mice as they do humans. This is due to our brain chemistry. I would also use multiple groups, 14 to be exact, 2 control groups and 12 experimental groups. 6 of these groups will be patients with early onset Alzheimer's, while the other 6 will be patients with late onset Alzheimer's. Out of both the early onset and the late onset groups, there will be 3 receiving drugs invasively and 3 receiving them non-invasively. All subjects will then be placed in the same living conditions, temperature, and have similar overall lifestyles. Biomarkers will be used similar to those found in previous research to assess the state of the disease, effectiveness of the therapy, identify toxicity and adverse effects, and assist in dosage selection. This research will be done in 4 phases. In the first phase, all test subjects will be given an MRI and an FDG PET to examine the amount of neurodegeneration before any possible mode of treatment. This stands as a comparative model for our experimental group as well as our control. Researchers will then divide each individual into its own quarters to ensure no interactions would affect the results. These individuals will be classified through a tag as well as through identification on their living quarters. Everyone will then be given its first dosage or placebo. This dose will be given at the start of the week through nanotechnology. Most likely, this will be through lipid-based carriers and organic nanostructures. The biomarkers and other scans will proceed at the end of the week and be compared to previous data. This will repeat until management of neurodegeneration has successfully occurred. We are saying 1 to 5 months. In phase 2, we will then lower, maintain, or increase the dosage given to each individual. This will determine the increased amount of a particular substance. We will then measure how neurodegeneration is affected, if this speeds up the process, and if the full dosage can be transferred through the blood barrier level. We would ultimately measure if this would cause toxicity within the body. Biomarkers will determine this. On the other hand, lowering the dosage would allow us to track how significant the dose amount is in managing the neurodegeneration. Ultimately, phase 3, the dosage will maintain at the same rate. This will be carried out for many months to ensure that all patients see similar results to other groups. This will allow for the correct dosage to be mass-produced and approved as the correct form of treatment. In this phase, we will also test the success of using different nanoparticles, some being organic, non-organic, lipid-based, or even metal-based. This would allow us to determine which materials translate for the best result. And lastly, phase 4. If the previous 3 stages are successful, researchers would be able to understand nanotechnology and drugs used to fight against mutations leading to neurodegeneration and the onset of Alzheimer's. By using technology to edit the genome and alter proteins and chemicals of the brain, we could later use what we know now on other diseases of similar symptoms. Ultimately, I believe that nasal delivery through lipid-based nanostructures will result in the most effective treatment technology option. This is most effective because the delivery of the drug is faster than through the invasive brain delivery and sends a larger amount of the anti-AD drug to the brain, meaning more of this drug would pass through the blood-brain barrier level. As shown through the map, the rate if treatment occurs through nanotechnology would majorly decrease the amount of Alzheimer's that we see in our country and our world today. So why should you focus on this? Why should we think about the treatment for this? Due to the competitive nature of many companies to find a proper cure, my research would allow our university to not only use cutting-edge technology, but allow for the university to be put on the map. With this information, companies will invest and fund more research, making this profitable for the university and build a name for researchers on our team. While all of this is rewarding, restoring what has been lost, a stable, sound mind would be the most significant accomplishment to all researchers dedicated to this study. Through your funding, we can be part of a project larger than ourselves, one that will not only help one, but millions. Thank you for your time and consideration. I am Hampton Hand.